Hello students. In the topic principle of inheritance and variation, today we are going to discuss about the sex determination in male and female different organisms. How is sex determined? Either it is male or it is a female. In its means we can say it is before some animals we can determine the sex before the fertilization. In some at the time of fertilization. And in some cases, what happened after fertilization, we can understand that this specific organism is developed into a female or a male. Okay. So first, here, what is given in NCERT, you have to see this one. The mechanism of sex determination has always been a puzzle before the geneticists, right? How they determine that the coming offspring, it is male or female in its embryonic stage. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insect. It is given here, carried out in insect. Why in insect only? Why not other organisms? That is a question you can ask. You can see here, it is given genetic chromosomal mechanism of sex determination can be traced back to some of the experiments which are carried out in insect, right? Carried out in insect. In fact, why insect? That I asked you, because in insects we have different type of sex determination we can obtain. As for example, when we consider the Drosophila, it shows XY type determination. When we consider the grasshopper, it shows at X0 type. When we consider the moths, so there we can see here there is a heterogamity of female, and when we see the honeybee, so haplodiploidy condition we can get there, right? As for example, here you can understand in case of insect, when we see the fruit fly, you know fruit fly means what? Fruit fly means Drosophila. So here it shows XY type of sex determination, XY type. Right, we can say female and male. This side it is female and this side it is male, right? And when we see the grasshopper, at that time we understand that it shows X0 male and XX female. When we see the moths or butterfly, we can say, right, butterfly, they shows just here, they shows heterogamity in male, heterogamity in female, and we can say here that is and type. In case of moth butterfly, this heterogamity in female, it is also present in birds and it is also present in moth, right? And when we see the honeybee, so we understand here N for male and 2N for female. Means here haploidy and diploidy condition is fine, right? In this way, here because different type of sex determination is present in insect, that why the insects are the best experiments for sex determination, right? In fact, the cytological observation made in a number of insects led to the development of concept of genetic chromosomal basis of sex determination. Henneking. First it is given Henneking. That is important science name, Henneking, right? This. In 1891, could trace, because Henneking is the first scientist who discovered the X chromosome, right? So he could trace the specific nuclear structure at all through spermatogenesis. Where? In spermatogenesis. This is very important. Spermatogenesis. Not in gametogenesis, not in oogenesis, but in spermatogenesis. In a few insects, and it was also observed by him that 50% of sperms, how many? 50%. 50% of the sperms, they received this structure. This structure means X structure, right? After spermatogenesis. Because we know here, mostly what is there? Mostly males are heterogamy. They show the heterogamity. They are made up of one X and one Y chromosome. Heterogamety means they produce two types of gametes. That's why it is called heterogamety. And when either it is male or female, when it produces only one type of gamete, then it is called a homogamety. As for example, mostly females, they are homogamety because they produce only one type of ovum. Ovum means gamete, that is X, right? But in male, X and Y is there, or in means we can in Drosophila. And when we consider the grasshopper, that means one contains X chromosome, another means no X chromosome is there, X0 type, right? That is given here. In spermatogenesis, 50% of sperm receive this structure, means X structure. We can say here receive this X structure, right? Whereas the other 50% sperm, they did not receive. They did not receive. We can see here this X, right? Because here why he used why in spermatogenesis? Because in spermatogenesis only heterogamety is present. Oogenesis, why did not he find it out this one? Because oogenesis means what? In the formation of ovum, 100% gametes they receive the X chromosome, right? Then he cannot determine whether they are 50% different or not, because 100% are same here. Whereas 50% sperm did not receive it. Hanking gave a name of this structure. This structure means this X chromosome structure called X bodies. He said them X bodies, right? But he could not explain the significance. What is the significance of this X body? What are they? He could not explain this one. So that is here we can see the who discovered the X chromosomes or who first see the X chromosome is Hanking. In which year? 1891, right? You understand here, he could not explain its significance. Further investigations by other scientists led to the conclusion that the X bodies of Hanking was in fact the chromosomes. Then after then the scientists came and they discovered that this X body is actually X chromosomes, right? And that is why it was given the name X chromosome, because he said them this is the X bodies, and that's why they give the name as X chromosome. He give the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of X0 type. X0 type means here the number of chromosomes is not equal in male and female, means in female n-1 chromosome is present when we compare with male, right? Suppose that male it has 17 chromosomes, that means female contains 18 chromosomes, one more, right? Because here 23rd chromosome is absent, not 23rd means sex chromosome is absent, X0 type. That means all the X bear an additional X chromosome beside the other chromosomes, autosomes. That means we can say autosome plus X, that means female, autosome plus X0 is O or 0 we can say, right? That means male. On the other hand, some of the sperm bears X chromosomes, whereas some do not. That's why if they are fertilized with sperms which bear X chromosome, so they forms female, and if they fertilize with the sperms which have no X chromosomes, they develop into male. Egg fertilized by sperm having X chromosome becomes female, and those fertilized by sperms they do not have X chromosome, they become the male. X0. As for example, grasshopper, I already told you. Do you think the number of chromosomes in the male and female are equal? No, not equal. Because female it has one more X chromosome, that means number of chromosome is one more than number of chromosome of male, right? Due to the involvement of X chromosome in the determination of sex, it was designated be the sex chromosome, right? What is there? Involvement of X chromosome in the determination of sex. That's why it is called as sex chromosome. Do you understand this one? And the rest of the chromosomes were named as autosome. This sex chromosome, another name is for allosome. Allosome or sex chromosome we can say, right? Another means autosomes, somatic chromosomes. Grasshopper is an example of X0 type of determination of in which male have only one X chromosome, right? with autosomes, whereas the female, they have, look, grasshopper is an example of X0 type of sex determination, in which the males have only X chromosome beside the autosomes, whereas the female, they have a pair of X chromosomes. Autosome plus XX, that is female. Autosome plus X0, that is male. As for example, grasshopper. Most of the insect, they determine through this, right, determination. Second, these observations led to the investigation of a number of species to understand the mechanism of sex determination in a number of other insects and mammals also, including human. We are mammals, right? Which type of determination is there? XY type of sex determination is seen where both male and female have the same number of chromosomes, right? But one thing is different here. Among the male, an X chromosome is present, but its counterpart is distinctly smaller and called Y chromosome. It is smaller and 3-arm chromosome. That's why it is called as Y chromosomes. Females, here, both males and females, they bear the same number of chromosome, 46 chromosome in us, right? And when we consider this Drosophila, it has also 8 chromosomes in both, right? Hence, the male and have autosome plus XY, while the female have an autosome plus XX. So this one is called XX and XY type of sex determination. In human beings and in Drosophila, and the name is fruit fly, the male have X and Y chromosome, whereas the female have a pair of X chromosomes beside autosomes. Here we read two types of sex determination, XX, X0 type and XX, XY type, right? Now third is there. In the above description, you have studied about two types of determining the mechanism, that is X0 type and XY type. You can write here, you can see it is XO type. We can see because O and 0, they are same symbols. 0 means absent, that's why we use O. XO type and XY type, but in both cases, male produce two different type of gamete. Because male produce two different type of gametes, that's why males are called as heterogametic. A, either with or without X chromosome, and B, some gamete with X chromosome and some with Y chromosome. 50% here, in case of X0, 50% they contain X chromosome, 50% they have no X chromosome. In XY condition, 50% they have X chromosome, 50% they have Y chromosome. That's why they are called as heterogametic, because two types of gametes are formed. Such types of sex determination mechanism is designated to be the example of male heterogamety. That's why it is called as male heterogamety, and I think you understand why it is called male heterogamety. Hetero means different, gamety means gametes. So because they produce, these means man or mammals, right? Drosophila. They, or second type is grasshopper, right? So they produce two different type of gametes. That's why they are called, that males are called heterogametic. And this sex determination is also called as male heterogametic. In some other organisms, as for example, birds. Bird is given here, not only bird, right? But we can see here in case of moths. Moths means like butterfly, reptiles. Then bird is also given here. Fishes, right? These animals, they shows female heterogamety. They shows female heterogamety. A different mechanism of sex determination is observed, right? This one. See the figure, we will see the figure, right, after reading this one. In this case, the total number of chromosomes is same in both male and female. Their number is same also. XY means number is same. This one is also number is same. Only two sex determination are there in which number is different. One is haploid, diploidy, as for example in honeybee, and second in case of moth. Moth means only one less chromosome is there. Both are 2n, but in male only one less chromosome is there, right? And haplodiploidy means male honeybee, they have half set of chromosome, means only n, half number of chromosomes are there, right? Here, but two different type of gametes in term of the sex are produced by the females. Who produce this one? By the females. That means here female heterogamety. Here what is it is said? It is called as female heterogamety. Because here female they produce two different type of gametes. Male means they produce only a single type of gametes, just opposite of XY and XY, right? In order to have the distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of female bird has been designated to be Z and W chromosome. In case of bird, because this the capital letters we are using for represent the chromosomes, it is actually on the base of the structure of chromosome. Because in humans or Drosophila, that means they have 4 arms are there, and we know X means what? X has 4 arms. Our Y chromosome is the smallest chromosome, and it has only 3 arms, and in Y 3 arms is there. So mostly we means effort to make a structure which is resembles with the chromosome structure, right? Means the alphabet which we are used, right? They are means not accurate, but okay, nearly the same structure of the alphabet. Z and Y W chromosome. In these organisms, the females have Z and 1 W chromosome. 1 Z and 1 means we can say 50% Z and 50% W chromosomes. Whereas males have a pair of same chromosome. Same means which one? Z chromosome beside the autosomes. Here we can say here this represents the female heterogamety, male homogamety, right? One thing is there, and their chromosome ZZ and ZW type of sex determination. So do you understand what is here? Right? Okay, first we read this one. We will complete this one, then I explain something other. Now sex determination in humans. Already you understand what is the sex determination in human. We use XX and XY type, right? That is given. It has already been mentioned that the sex determining mechanism in case of human is XY type. Now we understand in human it is only XY type of sex determination. XY type. Here what is out of 23 pairs of chromosomes which are present in us, 22 pairs are autosomes and 23rd pair is called allosomes or sex chromosomes. So 22 pairs they are exactly same in both male and female, because they are responsible for the formation of different part of our body, right? These are the autosomes. These are called autosomes. 22 they are autosomes, and the last one, that is 23rd, a pair of X chromosome are present in female, that is allosome or sex chromosome, and in the presence of X and Y chromosome are determinant of the male characteristics. That is third one, because it is responsible for the sex determination. That's why they are called as sex chromosome or allosomes. During spermatogenesis, right, among the males, 2 type of gametes are produced. 50% of the total sperm they produce only they contains X chromosome, and 50% they contains Y chromosome. That is given here. 50% of the total sperm produced carry the X chromosome, and the rest 50% has Y chromosome beside the autosomes. Autosomes means it is present in female and male both, right? Now females, however, produce only one type of ovum, because they have only XX, both chromosomes are same, only one type of ovum, that is which contains X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. 50-50% are there. So anyone can fertilize, either X or Y can fertilize, right? In the case of ovum which is fertilized with sperm that carry X chromosome, it develop into female, and sperm which carry Y chromosome, it fertilize, then it develop into a male. The zygote develop into a female, XX, and the fertilization of ovum with Y chromosome carrying sperm results into male offspring. Here what is there? Why it is given here? Because here the who is responsible, that type of sperm which fertilize the ovum is responsible for the male or female, right? Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. And it is also evident that in each pregnancy, there is always 50% chances or probability of either a male or a female child will be born, right? It is unfortunate that our society, this time we are okay, this time we are literate, right? And we understand everything. But before, I think some decades, what happens? They only blame to the women, right? That is one. Women are blamed for giving birth to female children and has been ostracized and ill-treated because of this false notion. Now we understand who is responsible. Male is the responsible. That sperm is responsible for the male or female child. And one last, because this one is XX, XY type, XX, X0 type, Z and ZW type of sex determination, right? And now haplodiploidy type of sex determination, and this example that is given in case of honeybee here. Not only in honeybee, right? Honeybee means it is a insect. So different insect they also shows this type of heterogamity. Oh, sorry, the heterogamity means what? They are haplodiploidy, right? What happens here? Because we know here male honeybee, it is developed through unfertilized egg. If egg is fertilized, it develops into female. If egg is not fertilized, then it develops into a male. That is, fertilized means what? It becomes 2n. Unfertilized means what? It is haploid only n. That is even here. The sex determination in honeybee is based on the number of set of chromosomes and in an individual receives. An offspring formed from the union of sperm and egg. Union of sperm and egg means what? Fertilized egg, right? As a female, it may be queen or worker, both are female, right? It is, and now queen or worker, it is depend upon their feeding mechanism. Because if it is the female, this female, if it is feed up upon royal jelly, right, it develops into a queen. And if it is fed by this is we can say that is honey cakes. Cakes means what is it? That is wax-like substances, right? Honey cakes. Then they develop into worker, right? And an unfertilized egg, which is not fertilized, it develops into male, or it is called as brood, by means of parthenogenesis. Because here fusion is not happening, fusion is absent, and we know when we read an botany section, right? Then what we read? Parthenocarpy, a fruit which is developed without fertilization. That fruit is called, or seedless fruit is called parthenocarpic fruit, right? And this process is called parthenogenesis. Here it is also because these males, they develop without fertilization. That's why this process is called parthenogenesis, right? Parthenogenesis. This means that the male have half number of chromosomes than that of the female, because it is unfertilized. That means it is one set is absent here. It is N only. The females are diploid and having 32 chromosomes. 32 diploid means 2n, and 2n means what? 32. That is even here. Diploid means what? 32, 2n, right? And that's why here only n is there. Here what happens? Because female 32 means how female get 32? Because we know fusion after n plus 1, n becomes 2n, means 32, develop into female, and because it is unfertilized, it has only 16 chromosomes. These are male, develop into male, right? And males have haploid that having 16 chromosomes, only 16 chromosomes. This is called as haplodiploid sex determination. What is called? That specific name is given, haplodiplo. Haplo means haploid, haploid means male, diplo means diploid, diploid means female, sex determination. right? And this process is called parthenogenesis. Develop male drone without unfertilized, without fertilization. Male develop without fertilization. That's why this process called parthenogenesis, right? And has a special characteristics feature such as the male produce sperm by mitosis. This is also very important because we know gametogenesis means meiosis is there, but at least this time we understand that all the organisms they produce gamete through meiosis. That is wrong, because when we consider the plant, so we know bryophytes, their bryophytes, they produce bryophytes, their chief, or we can say male plant body is gametophyte. Gametophyte is haploid, already haploid. So gametophyte produce gametes through mitosis in case of bryophytes, right? And here we understand that because males are already haploid, so they also produce sperms through mitosis. Because meiosis, if meiosis happens, what happens? Reduction of division. Reduction means now the number of chromosomes become 8, and that is not possible in nature, right? Only meiosis. Meiosis means equational division. Right? Means they produce an haploid. That's why they produce haploid, right? They do not have father and thus cannot have sons. They do not have father, then thus cannot have son. Why they do not have father? Because male they produce through unfertilized. Unfertilized means here no male sperm is used for fertilization. A sperm is absent. Sperm come from father. When sperm is absent, means father is absent. So they have no father. And they have no sons. No son, why? Because they produce sperms, and this sperm, it fertilize. If it fertilize, so this egg develop into female, not in male. Egg which is unfertilized, that develop into a male. That's why it is given they cannot have son. They have no father, no son. But one thing is there, they have grandfather and grandson. Why we said them grandfather and grandson? Because mother developed through fusion, fertilization. Mother developed, that means we can say mother has father. They have daughter, they have mother, they have daughter, but they have no father, no son. That is here. Because they have mother, the mother developed through fusion, that means they have grandfather, right? Grandfather means, okay, I can explain you here, right? Why it is called grandfather. They do not have father and thus cannot have sons, but they have grandfather and can have grandsons, right? Okay. How is the sex determination mechanism different in the birds? How? Because here in birds, how it is different? Because in birds, it is female heterogamy. It shows female heterogamy, right? That's why different. And mostly other animals means male heterogamity. Is the sperm or the egg responsible for the sex of chicks? Who is responsible? Sperm, because it produces only one, that is ZZ chromosome. ZZ, only one type of sperms. That means it is not responsible. Who is responsible? This egg is responsible because it 50% Z chromosome and 50% Y chromosomes are produced by 50-50%, right? 50-50%, that we can say 1:1. So who is responsible? This egg is responsible for sex of the chicks. Okay, here it is given parent, this is the female, and this side male is there, right? When you see after meiosis, what happens? 16 chromosomes are, these two gametes are formed. Here two gametes, one gamete, and this is the second gamete. 16. If it is unfertilized, F1 generation means unfertilized, no fertilization in it. Means in the absence of fusion, they develop into male, right? Because no fusion is there, so they develop into male, right? In male, what happens? Look here, male, because they have n number of chromosomes, mitosis, so they produce sperm through mitosis. They produce sperm through meiosis. That is the first difference, right? 16 chromosome. Female means 16 chromosome. Now what happens? 16 another, right? And 16 this. When they fuse with each other, then after fertilization present, right? So fertilized egg develop into female, we can say here fusion occurs, fusion present, right? And here fusion absent. So they develop into male and they develop into female. You understand? Because this sperm come from here, come from father. This sperm, it is the mother's sperm, right? Mother's sperm and father's sperm, that means develop into female. Female daughter, they have a father. This one becomes father, right? But here no fusion means what happens? No fusion means only mother is there, but no father. Okay, before going to mutation, first a structure is given. We will discuss about the structure and then yes, you can see here XX and XY in humans. That here XX and XY in Drosophila case. Drosophila, this is heterogamity. Male means heterogamity, female means homogamity is there, right? These both, they shows heterogamity in case of male, right? And female means they shows homogamity. Homogamity, same chromosomes are there. Here two different chromosomes are there. These two types of species. Second, it is also Drosophila, right? Now, but when we see this one, these are chicks. It has two different Z and W. That means what? This female, it shows heterogamity and it shows homogamity, right? That one, because both have XZZ type homogamity. That is the main difference which is given here. Determination of sex by chromosomal differences A and B both in humans and in Drosophila. The female has a pair of XX chromosome, that is homogamy, right? And the male has XY chromosome, that is called heterogamity. I'll tell you, male means heterogamity, right? This one, and female means homogamity. This is the female, this one is the female, homogamity, right? Compositions. In many birds, female has a pair of dissimilar chromosome. You can see ZW type, and the male have two similar ZZ and ZZ chromosomes as you see here. That's here only what you see here. We can understand that in case of Drosophila and in humans means all mammals, they have male heterogamity and female homogamity is there. Male produce two different type of chromosomes and female produce only a single type of chromosomes. In case of birds, in case of moth, reptiles, they shows just opposite this one, means female shows heterogamity, Z and W type, means two type of gametes, one is Z type, another one is W type. But male, it produces only a single type of sperms, that is, we can say Z and Z type. So they shows the homogamity in case of male, but heterogamity in case of female. I think you understand. Here, what we read in this chapter. Sex determination, right? When we consider the sex determination on the basis of fertilization, first means on the basis of fertilization. Then we have three types of sex determination, right? Three types. How many types are there? Three types. Which three types? First one is called, because it is determined before fertilization, so it is called as progamy. Progamy means before fertilization, because it is a progamic, right? Before fertilization. As for example, in case of male honey bee, without fertilization they develop into male. We understand no fertilization occur, that they develop into male. That's why progamic means male. Now second is our syngamic. Syn means at the time of fertilization. Syngamic at the time of fertilization. Mostly means we can say here most of the organisms simply. Organisms. Human also here. If it is fertilized with X chromosome means female, fertilized with Y sperm, that means male, right? And third is means what happens here, it is after the fertilization. When it is determined after the fertilization, then it is called as epigamy. Epigamy, after fertilization. After fertilization, as for example, here female, we can say female honeybee, right? So these three types of sex determination on the basis of fertilization. When we consider the fertilization, this is not given in NCERT, but sometimes it is asked a progamic type of sex determination, means before fertilization, right? So this time you have to understand what is the meaning of progamic, syngamic or epigamic. Progamic means before fertilization, syngamic means at the time of fertilization, fusion of fertilization, and epigamic means after the fertilization. Okay. Now next point is our second is called here, because chromosome, right? Chromosome base of sex determination, or we can say because sex chromosome are also called as we can say allosomic. Here I write here allosomic or sex chromosome, right? Allosomic determination of sex. This is given our NCERT. How many types of there? Only we have to revise, right? And we know first some information I want to give you. Then after we will discuss this, right? As for example, first is question here. I write here allosome. Allosome. Allosome is what? Sex chromosome. First question is there, who gave this term, allosome or heterosome? Allosome term was given by Montgomery. M-O-N-T, Montgomery. This term is given by, first he used the term. Which term? Allosome. Montgomery, he used this term, right? Only not this term, heterosome and allosome both is used here. Heterosome or allosome. These two terms, it is used by Montgomery, right? This side. Second thing is here, in our NCERT it is given, who first discovered the X chromosome? X chromosome were discovered by Hanking, right? And figure also it is given in 1891, and he said them this X chromosome as X bodies, right? Second is there, who first discovered the sex chromosome? So this is who sex chromosomes are first discovered by, here we can write here first discovered by, who discovered? McClung. His name is McClung. McClung in grasshopper. Right? McClung in grasshopper. He first discovered the sex chromosomes. So this is the history about the discovery of sex chromosomes, right? And one more, who proposed this sex chromosome determination of sex? So he is our actually Stevens, right? Who? Wilson and Stevens. Wilson and Stevens first proposed the theory of sex determination, right? Chromosomal theory of sex determination, actually. Theory of sex determination. This theory is proposed by first, who proposed this one? Wilson and Stevens. So this is some historical events which are important for sex determination, right? Now what we read in this chapter here, we can say here, how many types of chromosomal type of sex determination? First we write here XX and X0 type, right? Second we read here XX and XY type. Third we read here, third means what? It is our, we can say, which one? Z and ZW type. Hetero, female, heterogamety. And last we understood here haplodiploidy type. These are the four types of sex determination which we read in this chapter. Which one? XX0 type means here number of chromosomes both are diploid. We can say both diploid means 2n, but number of chromosomes are unequal. Unequal chromosome number. Right? N-1 in male. That is, as for example is grasshopper. Second is XN, XFI type. It is both diploid. 2n equal number of chromosomes, right? Example means Drosophila, humans, mammals other, right? ZZW type means both diploid. 2n. Number of chromosomes are also equal. That is, number of chromosomes is equal, right? Right? Equal. And as for example, bird, moth, reptiles, fishes, right? All these are diploid, but what is the difference? Difference means here these two, they shows male heterogamity, right? But this one, it shows female heterogamity. Female heterogametes. Now this haplodiploidy type means here we understand this one is male and this one is female, right? Female means male means what? N. Female means what? 2n, right? When they crossed, right? What happens when they cross? Cross means sperm from this side. So it produce, okay, first I write it. It produce mitosis. Through mitosis it produce sperms, and through meiosis it produce gametes. Two gametes are formed, 16N and N. Only one gamete, that is N, is produced, right? And when it is crossed, then what happens? If they crossed, they fused, they forms female to N. That one is female, right? It is female, daughter. And if it is unfertilized, it develop into, without fertilization, it develop into N, that is male, right? Because here father is absent, so they have no father. No father. Why not father? Because as no fertilization is there, no fusion of sperm. That's why no father, right? But they have father because fusion happens. This is the daughter. It has father because fusion is present. Fusion with male. Fusion is present here. That's why it is father. That's why we can say they have no father and they have no son, because here no son is there. Only daughter is there. So that means it has only daughters, but no son. Right? They have no father because no fusion is there, right? Now this is also due to the it has father and mother because it produced through male and female, right? Gametes, because due to the fusion. That means we can say this one is the grandfather of this son. You understand? This son has mother and mother has father. So this one becomes grandfather. So they have grandfather, these sons, right? They have grandfather and they have no son. Look, they have grandfather, but they have no father, and they have also no son. Because no son, why no son? Because no son developed through unfertilized egg. You understand this one? Why it is called haplodiploid type means what? Here number of chromosome, we can say number of chromosome in male, it is only N, that is haploid, means half, means unequal. We can say unequal, right? But number of chromosome in female, it is 2n, that is diploid, right? Means double, right? And that's why it is called as haplodiploidy type. Do you understand why it is called haplodiploid? This one is called haplodiploidy ZZW and XX. This we read in our NCERT book, right? So this is the another type of, it is called chromosomal theory of sex determination, which was given by, who gave this one? Wilson and Stevens. Okay. Now another one is also there, cytological. Cytological means bar bodies, through bar bodies. Cytological. Cytological sex determination means what? Here, this one is called Laien hypothesis. Laien hypothesis. Here what happens? Here this X chromosome is bar bodies. X bodies means it is also called as BARR, right? Bar bodies. bar body, right? Bar body means a dark spot is there. It is when we see this through microscope, we see a dark spot, right? Now here we can understand that means in case of male, this bar body means number of bar bodies, simply we write here. Number of bar body is equal to n minus 1 X chromosome, right? X chromosome, right? As for example, in case of XY, number of bar body means 0 bar bodies. That means what? It is male, right? It is represented as male, right? And if it is XX, then what happens? Only one bar body. It develops into female, normal. Right? Sometimes what happens in case of trisomy, for example, XXX, then what happens? 2 bar bodies. Here what we said, this is the trisomy. We can say trisomy, and this is the disorder. So through the number of bar bodies, we can understand it is male or it is female, or it has some this genetic disorder also. We can understand through this one, right? So this is called cytological sex determination. Okay, I think you understand. Now here next point is our mutation. Mutation. Mutation is the mutation means what? Mutation. Mute, actually mute means what? Mute means silent. Here mutation means what? Mutation. Who gives this first means who gives this term mutation? Hugo de Vries. Hugo de Vries. Hugo de Vries is called as father of mutational theory. Hugo de Vries. Which one? Hugo de Vries. Right? Mutation is a phenomenon which results the alteration of DNA, or we can say alteration of chromosomes. That mutation, right? Who give this term? Hugo de Vries, right? Hugo de Vries. In which plant he experiment this one? In evening primrose. Evening primrose, right? Is a phenomenon. That means evening primrose. What? Mutation means what? Suddenly we can say here, mutation is referred as sudden change which is inherited, in offspring. Right? Sudden changes which inherited in offspring. That is called mutation. It is given a phenomenon which results in alteration of DNA sequence and consequentially results in change of the genotype as well as the phenotype of the organism. Because if the DNA sequence means what? Nitrogen base or nucleotide sequence is changed, then what happens? The sequence of amino acids is also changed. Because DNA is responsible for formation of messenger RNA. Messenger RNA forms the polypeptide chain. Polypeptide forms the proteins, which are the enzymes. Enzymes are responsible for phenotype expression, right? That's why if a DNA sequence is altered, then what happens? Genotype is altered, and because genotype is altered, that means what? Expression is also altered. Expression means what? Phenotype is altered. In addition to recombination, mutation is another phenomenon that lead to variation in DNA. Recombination, right? Mutation is another phenomenon that lead to variation in DNA. Mutation means, okay, simply this word comes from mute. Mute means what? Some part of the DNA becomes mute. Mute means what? It cannot express anything. According to Hugo de Vries, but here what happens? In addition to recombination means what? Crossing over. Crossing over is another thing. Crossing over means exchange of genes. That then after what we get? We get variations. And mutation is another. Another means what? Some parts are deleted, some parts are added, or we can say DNA is totally altered, which is not a normal, which is an abnormal phenomenon, which is responsible for variation. Also, we can say mutation also responsible for variation. Recombination also responsible for variation. But recombination, it is a natural phenomenon. But mutation, what happens? It is a sudden phenomenon. Sudden, right? Recombination means not a means we cannot say recombination means deletion or addition. There is some alteration which is harmful. But mutation means mutagens are also responsible for mutation, but recombination, it is a natural phenomenon in which exchange of genes in case of meiosis, prophase first, crossing over, right? The recombination happens. That's why I keep on. In addition to recombination, both are responsible for variations in new offspring. But in addition to recombination, mutation is another phenomenon that leads to variation in DNA. Recombination in sexual reproduction, what happens at the time of prophase? First, we know crossing over happens, and crossing over means what happens here? Exchange of genes are there. When genes are exchanged, then new offspring, it has variation. Mutation means something is delayed, something is added, or anything is there, right? Then what happens? It is also leads to variation. So both leads to variation, right? But both phenomena is different. As you will learn in chapter 5. In chapter 5 already we will learn this one, right? I tell you the detail study. I will tell you detail when we briefly about the mutation, different type of mutation. We will discuss the next chapter, means 5th chapter, right? This is our 4th chapter. We will read this one, chapter 5. One DNA helix runs continuously from one end to the other in each chromatids is a highly supercoiled form. That is, this supercoiled form is called solenoid form, right? Therefore, loss, deletion or gain, insertion or duplication. Look, loss means what? Deleted part. If some part is deleted, some part is cut off, that means called deleted or loss, or gain or some another part come and add. It's called insertion or duplication of a segment of DNA result in the alteration of chromosomes. And since genes are known to be located on chromosomes, that means what happens? Alteration in chromosomes results alternation or abnormalities or aberration of chromosomes, right? Chromosomal aberrations are what? Here, look here. This aberration means what? Change in the structure of chromosomes, right? Aberration means what? Change in the two types of mutations there. We can say chromosomal mutation and gene mutation, right? First thing. Now, chromosomal mutation is also two types. First one is called change in the number of chromosome, right? Change in the number of chromosome set. Number of chromosomes, okay, it is called ploidy. Ploidy is also two types. It is euploidy or aneuploidy. If set of chromosome is changed, then it is called euploidy. And if number of chromosome is changed, it is called aneuploidy. We will read this briefly in the next chapter, right? Second is chromosomal aberration. Aberration means when the chromosome shape is altered, that is called chromosomal aberration. It is also deletion, inversion, duplication and translocation, right? Frame shift mutation, point mutation. These are the type of which one? Aberration. Aberration means what? Four types. Which one? Deletion, inversion, duplication and translocation. Deletion means in a single chromosome. Inversion means single chromosome. When we consider duplication means between homologous chromosome, and translocation means between non-homologous chromosome. That's why translocation type of chromosomal aberration is also called as illegal or illegitimate mutation, right? And gene mutation, two types we will discuss here, that is substitution or point mutation and frame shift mutation, okay? In addition to the above, mutation also arise due to change in a single base pair of single base pair. If a single nucleotide change, then it is called as point mutation. Point mutation, because a specific point it is changed, right? A classical example of such a mutation is sickle cell anemia. Sickle cell anemia, what happens here? Only a single nucleotide is replaced. Which one is replaced? A is replaced by U. In case of messenger RNA, when we see the messenger RNA, right? In messenger RNA, GAG, that is the sixth genetic code, right? genetic code, it is GAG, right, which is responsible for the amino acid. Which amino acid? Phenylalanine, right? This one is this phenylalanine, or we can say GAA. A means what? Adenylate acid is replaced by uridylic acid. In case of I am telling about the messenger RNA, not DNA, right? Uridylic acid. It becomes GAG becomes GUG, and GUG is responsible for valine, and that makes the all difference, right? We can say if amino acid, which amino acid? Phenylalanine is replaced by valine, now sickle cell anemia is found. Or we can say nitrogen base, which nitrogen base? A, adenylate acid. Nitrogen base adenylate acid is nucleotide, right? It is replaced by uridylic acid. U means uracil. Then what happens? Sickle cell anemia is found. This is the sixth genetic code of beta chain, right? Because hemoglobin, we know hemoglobin is what? It is made up of, globin protein is made up of four polypeptide chains, 2 alpha and 2 beta. Sickle cell anemia means beta chain. The sixth genetic code of beta chain, it contains GAG in case of normal hemoglobin or normal RBC, but this GAG is replaced by GUG, right? Actually, G is okay. Which nitrogen base is replaced? A is replaced by U. Then it becomes GAG, becomes GUG. And GUG, it is responsible for valine, and that makes the difference. Okay? That is different. Deletion and insertion of base pair of DNA. Deletion, either one is deleted or two is deleted, or addition, one add, two add. Then what happens? The total backside that is a sequence of nitrogen base is totally changed. That's why it is called frame shift mutation. Frame shift mutation. Frame shift mutation is okay. If one nitrogen base change, total frame shift is changed. Say two are changed, total is changed, right? When one replaced, no change. Replaced means point mutation, only a single point. Frame shift means total frame is changed, right? So you have to see the chapter 5th where I detailed, where we are discussing detailed, right, about the different type of mutation. Okay, the mechanism of mutation beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce the mutations, right? So factors which induce the mutation, there are called as mutagens. These are referred to as mutagens. Ultraviolet radiation can cause mutation in organisms. It is also mutagens. Okay. So today we finish the mutation, right? And next lecture we will discuss about the genetic disorders. We will finish this chapter, and this symbol, some symbols are also given here. We will discuss it. If you have any doubts, you can ask. Okay. In order, thanks.